continuing our journey with DNA. So last week we discussed DNA structure. This week we're really getting into, we're getting into how we go from DNA to a protein. The central dogma of DNA is DNA to an RNA to a protein. And we're going to discuss the processes that occur to get us from DNA to RNA and then to a protein. And this is the foundation, the basis of life. So I need you guys to bear with me because this can get kind of complicated. So I'm going to give you some assignments to really work through this and try to help you understand it. But we need to really work through. So we've talked about protein production being done in ribosomes, right? And the rough ER has proteins, so it makes proteins and they go into the ER and they get transported and modified a bit and squeezed off into vesicles, which then go into Golgi apparatus, which then will be modified and shipped out to where they need to go, right? And that's actually showing you bloodstream because proteins can be shipped out to bloodstream. This is just a quick review of DNA replication. When we are forming new DNA, we use the 3 to 5 prime template strand. Okay, DNA polymerase comes in and adds these nucleosides that become nucleotides. We use ATP for energy. Okay. And we have our base pairing rules. C goes with G, A goes with T always in DNA. We're going to learn some new ones this time. The DNA strand will only grow from 5 prime to 3 prime. All right. And then it's showing us our energy again. And then DNA is made from 5 to 3 prime, always. And then we're kind of looking again here at the process. We split up. We have our leading strand here. We have our lagging strand here. Our leading strand is the 3 to 5 because we know DNA can only be built from 5 to 3. Okay. And again, we're looking at the whole process of DNA replication. And that's, again, when we want to replicate DNA. And when we talk about cell replication or cell division, we're going to talk about why we would need to do that. Now, on the lagging strand is the other strand, right? We see that we have an issue. We can't string them together, right? There's no energy to bond right there. So then what we do is we make these Okazaki fragments. Okay, and it gets strung together with this ligase here, right? There we go. So again, that is DNA replication. So here we're taking a look at a bunch of letters, which we're going to be very familiar with, but these are the genetic code. The genetic code is just a series of 4 repeating letters, and these letters are our nitrogen bases that are in the middle of the DNA. Okay. Where are the genes? We're going to talk about genes today and how they're read. Okay? All right. All of these questions that we are going to answer. So like I mentioned before, the central dogma is the flow of genetic information in a cell. So how do we move from DNA to proteins? So here we've talked about DNA to DNA through DNA replication, correct? Well, we need to get to DNA to RNA through a process called transcription. And I want you to think of literally to transcribe something, what that means. Then we go from RNA to a protein through translation. Again, think of it, what to translate means. And that protein codes for a trait. Okay. All right, so let us first talk about RNA. We've talked about RNA briefly when we talked about the structure of nucleic acids. RNA carries genetic information. But instead of deoxyribose, it has a ribose sugar. It has nitrogen bases, but instead of having a T, a thymine, it's got a uracil. So before in DNA, where A paired with T, we now have a U for RNA because RNA does not have a T. And we'll talk more about that. C and G still go together. It's single-stranded, so we're looking right here at a comparison of DNA and RNA. It's single-stranded, and there's a lot of different types of RNA. Messenger RNA. mRNA is messenger RNA. tRNA is transfer RNA. rRNA is ribosomal RNA. And siRNA is something that we're going to talk about at the end. Okay? So we're going to talk about DNA. So DNA gets turned into RNA. The genetic code DNA gets turned into RNA through a process called transcription. Okay? What is transcription? Transcription is when we take the DNA nucleic acid language and transcribe it to an RNA nucleic acid, okay? This occurs in the nucleus DNA cannot leave the nucleus, RNA can. So this is a very important process to get the genetic code eventually out of the nucleus to the ribosomes where proteins need to be made. All right, so here we have transcription. And transcription is making RNA. That's what it is. We have a transcribed DNA strand, which is our template strand. We have an untranscribed DNA strand, which is our coding strand. And that will have the same sequence as RNA. Okay. The synthesis of complementary RNA strand occurs in a transcription bubble. And the enzyme responsible is RNA polymerase. Okay, so this is a big RNA polymerase. So I want you to take a look here. We're taking a look at the 5' 5 to 3' strand and the 3 to 5' strand. So let's talk about these. The 3 to 5' strand is our template strand. The coding strand is the 5 to 3. So please remember, the template strand is what we're using to make the mRNA. The mRNA will have the same code as the coding strand. mRNA is built 5' to 3'. Okay, so this is a pretty important concept for us to understand. So really quickly, we're going to talk about transcription in prokaryotes and kind of just compare prokaryotic and eukaryotic transcription. Transcription in prokaryotes starts with initiation. The RNA polymerase will bind to what we call a promoter sequence in the DNA. And the role of the promoter is to give us a starting point. Where do we start reading it? The template strand, it's which strand we want to read, and the direction of the DNA. And the DNA is always read 3 to 5, and we build RNA 5 to 3. All right, this is showing you our promoter sequences. We're reading the DNA 3 to 5, okay? All right. Then we have elongation. The RNA polymerase copies DNA as it unwinds. Okay, they roughly about 20 base pairs at a time. There's 300 to 500 bases in a gene for prokaryotes. And again, we're building the RNA 5 to 3'. I keep saying that. You're going to see that over and over and over again. So they have a simple proofreading that we don't, because mRNA has such a short life, they don't edit mRNA. Sorry. And then termination during transcription, RNA polymerase stops at a termination sequence. Okay. Now we're going to spend most of our time discussing transcription in eukaryotes. So really, I said this earlier, DNA can't leave the nucleus. Okay, so the difference between prokaryotic and eukaryotic genes is this. Prokaryotes have DNA in the cytoplasm, it's a circular chromosome, and it's naked. And they don't have what's called introns. Introns are junk DNA, non-coding in between the sequence. We call them junk DNA. I personally don't think they're junk. I think that we just don't know what they do yet. Eukaryotes have DNA in their nucleus. They're linear chromosomes. The DNA is wound up around proteins. We have introns, which is junk DNA, and exons, which is coding sequence. And that says that right down here, and I believe that's something you need to know. The introns come out of eukaryotic genes, but we're going to talk about that after post-transcriptional. Okay. All right, so transcription in eukaryotes. There's 3 RNA polymerase enzymes. The one that we are worried about in transcription is RNA polymerase 2. It transcribes our genes, our DNA, into mRNA. All right, so we're going to talk about initiation of transcription in eukaryotes. There's an initiation complex, which consists of transcription factors, which are typically proteins that are going to bind to a promoter region, which is upstream of the gene. Sorry. So if we're taking a look at a starter point here, we have a template DNA strand. So we're going to say, here's our gene, here's our promoter. Okay. These guys turn on or off transcription. Typically, the promoter is what we call the TATA box, T-A-T-A. And if you remember back before when I was talking about DNA structure, the TA is a good starting point for us to pull the molecule apart because there's only 2 hydrogen bonds there. It's a recognition site for the transcription factors. So these transcription factors know they have to come in and bind, and then we're going to have all these transcription factors bind, and we know that RNA transcription is going to start right here in this transcription initiation complex. Okay, so the post-transcriptional processing is really important. After transcription occurs, right? So we have our RNA polymerase is going to come in and transcribe the DNA in the correct way, right? We're building from 5 to 3, which means 3 to 5. Again, I keep saying that. You're going to see it multiple times. Okay. All right. Post-transcriptional processing is really important. After we make the initial mRNA from our DNA strand, we have what's called our primary transcript, our pre-mRNA. So we just came in and we just put all the base pairs together. So we have A, if there's an A for DNA, there's going to be U. If there's a T, there's going to be an A. If there's going to be a G, there's a C, and so on, right? So we build this long pre-mRNA, and we call that our primary transcript. It needs work. It needs a lot of work because this mRNA is now going to carry the genetic information out of the cell. Okay, so we do what's called mRNA processing to make our mature mRNA. So the first thing that happens is we edit out the introns. The mRNA gets spliced by what we call spliceosomes, and they go ahead and they cut out these non-coding segments here called introns. So if you take a look here, coding. There. This is going to protect, we also need to protect the mRNA from the enzymes in the cytoplasm, because the second that this guy goes out of the nucleus, it's going to be attacked by enzymes and lysosomes. So the first thing we do is we add what's called a 5' cap on the 5' end. We add a cap. And then we add what's called a poly-A tail. We add a bunch of adenine nucleotides on here. And what's going to happen is enzymes are going to come in, they're just going to chop away at these tails. But it's not going to affect this coding region of the mRNA, which is the important, it's like the gold here. It's like, this is the information we want. Same here. It's going to protect this segment, this coding segment, right? So our introns get spliced out here, and we go from this length to this length. And then we add a 5' cap and a poly-A tail. Okay. And this is just showing you our eukaryotic DNA, our mRNA transcript, and then our mature RNA. All right. The splicing has to be accurate. There's no room for mistakes. A single base added or lost throws off the entire reading frame. And you guys don't know what that means right now, but it's going to be important in a little bit. So they're showing you here, right, is our reading frame. And this isn't relevant to us yet because we don't really understand what it is, but we're going to be talking about that next. All right. So these are our splicing enzymes. They're called snRNPs. They're small nuclear RNA molecules, okay? And they come in here. They're little proteins, and then we have spliceosomes that are several of them, and they recognize and splice out a specific sequence. There. And literally cut and paste. And it's just showing you how this is done. All right. So that ends translation. So just so you know, DNA... Let's just recap. DNA gets turned into RNA. DNA gets read, okay? The genetic code is turned into a pre-mRNA. The pre-mRNA is processed and introns are sliced out. 5' cap is added. Poly-A tail is added. Now the mRNA can leave the nucleus with the genetic code, and it could be safe. Where is it going to go? Well, what creates proteins? Ribosomes create proteins. So let's talk about what happens next. So we just got through the DNA to RNA through transcription. Now translation is when we go from nucleic acid to amino acid. And remember, amino acids are the subunits that make up proteins. Okay? So we're translating from nucleic acid to protein. So as always, we're going to start off talking about translation in prokaryotes. They don't have a nucleus, okay? So transcription and translation are simultaneous in bacteria. DNA is in the cytoplasm, so there's absolutely no mRNA editing, okay? Ribosomes will just read the mRNA as it's being transcribed. Okay, the differences between prokaryotic and eukaryotes is the time and physical separation between the processes. It takes eukaryotes about an hour from DNA to protein. Okay, and we also have RNA processing. So you're looking at a prokaryotic cell. This happens very quickly, happens a lot slower in eukaryotes. All right, so here I just want to take a look at what we're seeing here. We have our genetic information here. This is our gene. This is DNA unwinding, okay? And this is the transcription process. We go to mRNA. Now we're going to go out to the ribosome and talk about this whole process. So DNA to RNA, mRNA leaves through the nuclear pores, okay? And it goes out to the ribosome where proteins are synthesized using the instructions that were on the mRNA, and that's through translation. So how much mRNA codes for proteins? So what we're looking at here is important, and I have you guys doing a practice example of this. Here's our genetic code. From mRNA, this is what the mRNA code is. So TAAUGC. Okay, we're just using base pairing rules. Remembering there's no T in mRNA. So wherever you would want to put a T, you put a U. And now we have to get down to, we're going to read this. I'm going to teach you how to read this and get into what our protein is. So these are all names of amino acids. These code for amino acids, and you're going to learn how to do that. So how do we get there, right? How can we code for 20 amino acids with only 4 nucleotide bases? That's extraordinary. All right, so let's talk about what codons are first. Codons are blocks of 3 nucleotides that are decoded into a sequence of amino acids. So a codon, we deal with codons in mRNA, is every 3 bases. Remember, these are nitrogen bases. So the 3 letters, that's one codon. This is another codon. This is another codon. Here's another codon. These codons, I'm going to teach you how to figure it out, all code for a particular amino acid, and you guys will learn how to figure that out. Okay. So the code, this is what we call the universal genetic code. And at the end of this, I'm going to teach you how to use it a little bit better. This is the code for all life. And what we have here are different combinations of bases, right? And you could see that it only codes for 20 different amino acids. This is the strongest support for a common origin for all life. The code is redundant. There's several codons for each amino acid. If you think there's 4 for valine, 4 here for alanine, 2, right? The third base is what we call a wobble, okay? An important thing to mention is that we always have a start codon. It's AUG, and it's the amino acid we call methionine. That's where we know to start reading the code, okay? There are also what we call stop codons. These stop codons, and you can see them up here, that means we are done translating. The protein is finished. It's actually quite interesting how this works out, okay? All right. So how are these codons a match to amino acids? So hold on, let's see what, we're going to come back to this, all right? So here we are again. Let's talk about how this happens. All right. We also need to understand that there is something called transfer RNA structures. They have like what's called a clover leaf structure. So take a look at this tRNA here. It has an anticodon at the end. This anticodon is to bind with the mRNA. This is how we find where to bring our amino acids. This is the site where they have amino acids attached to them. So these little tRNAs carry amino acids with them everywhere, okay? And this is their 3' end. And here's their anticodon. And this is what we're looking at here. tRNAs are going to bring amino acids over to the mRNA. Okay. Again, we need to talk about the structure of ribosomes very quickly. Ribosomes facilitate the coupling of the tRNA to the mRNA codon. And there's a lot of debate, are they an organelle or are they an enzyme? Since we already know ribosomes produce proteins, okay? The ribosome structure has 2 components to it, 2 subunits. This is our large and this is our small. And what happens is the mRNA will just be sitting here and the ribosome detaches and just attaches to it, okay? So we have different sites on them. So we have an A site on, and that's the part that holds the tRNA next to the amino acid. it holds the tRNA carrying the next amino acid that's going to be added to the chain. Our P site, our peptidyl tRNA site, it holds the tRNA that's carrying the growing polypeptide chain, and I'll show you guys this all together in a minute. And then we have an E site, the exit site, and that's where the empty tRNA, after it attaches its amino acid, leaves. It's an exit site. Okay, so there's the A, there's the P, there's the E. So what happens here is our tRNA. It binds to our mRNA. Here's our mRNA code, the start codon. The tRNA says, oh, I know I need to go here. Here is the anticodon, UAC, base pairing rules. It's carrying its little amino acid here on the growing peptide site. Okay, so let's talk about building the polypeptide. We have initiation, where it brings together the mRNA, the ribosome subunits, and the initiator tRNA. Then we have elongation, and that's adding amino acids based on the codon sequence. And then termination, which is our end codon or our stop codon. Sorry, I went way too fast there. So we take a look right here, okay? We have our EPA site. We have the methionine fines, that's our start. We have the next one coming up, and what happens is the ribosome kind of ratchets down the length of the amino of this mRNA chain. So what happens here is we have a bond that forms between these two guys, and we know that's dehydration synthesis putting these amino acids together. We've talked about this at great length, dehydration synthesis occurring, okay? So as these two connect, now the ribosome moves one over, and this empty tRNA is going to go find more amino acids, and it's going to continue to grow here. Once we get to a stop codon, a UAA is a stop, something called a release factor comes, and the ribosome releases, and the protein is made. Goes into the ER, gets transported to the Golgi apparatus, and we have this whole process occurring here. This can be really confusing to really understand, so I'm going to be showing you guys a little animation afterwards. So again, it says, can you tell the story? So we have DNA. Here comes our RNA polymerase, okay? And then we have our mRNA being made. There's our pre-mRNA, which then turns into a mature mRNA. Oh, there's our exon and our intron. 5' cap is here. Poly A tail is here. So this is our mature mRNA leaving through the nucleus, the nuclear pore. Okay, it comes out and it gets attached to by the ribosome. Okay, there's our ribosome. Okay, there's our A site, there's our P site, there's our E site, and we see the ribosome moving down the mRNA chain, continuing to build its amino acid. Okay. All right. There's our polypeptide. All right. So genes, defining a gene is problematic because one gene can code for several protein products. Some genes code for only for RNA. Two genes can overlap, and there are many other complications. So genes are difficult to define, but they are coding regions. The human genome has 3.2 billion bases. Okay, I just want to show you the transcriptional unit of a gene. So we had talked briefly about some of these. We're talking about the promoter region here. Okay. Our transcription start here, translation, here are our exons, here are our introns, which get spliced out eventually. So this is our transcriptional unit. These are all the components of that. We have our pre-mRNA, and then you can see we took out the introns, and we're going to add a poly A tail and a 5' cap, and then call it a day. Our transcriptional unit. All right, let's see what else is going to pop up here. There we go. Here's my poly A tail and our 5' cap. All right, so I wanted to put this in here because I have you guys working on something where you have to know how to use the universal genetic code. If we're looking to figure out which amino acid goes where, which you're going to be required to know, this is how we do that. We use the mRNA code. So if I have the code UAC, I'm going to find my first letter is U, so I know I'm in this row. My second letter is A, so I know I'm in this box. My third letter is C, so I know I'm at UAC, codes for TYR, which happens to be tyrosine. Let's say I do CAC, right? So C is my first letter, we're in this row. A is my second letter, I'm in this box. C is my last letter, right here. and I know it codes for HIS, histine. Okay, it's really important that we know how to do this. We're going to practice. So really quickly, see if you can figure out what GCG codes for. GCG. Pause it, figure it out. I'm going to pretend like you paused it there, and it codes for GCG, alanine. Okay. I'm going to watch this quick video with you.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Interesting stuff, guys. All right, let's go back to here. So if you have any questions, make sure you ask. I know that was a lot of information, but we will make sure that we understand it. Okay? All right.